Hello, hello. It is great to be here. It is great to be ready to learn and teach some biology. So I am very, very happy to be here. And who do we got here? Who do we got here ready to learn some biology? Can you please just let me know where in the world you are joining me from? Because I'm here in Berkeley, California, and I am just very excited to be here with people around the world. So let's see. We've got someone from New York. That's fantastic. I was once from New York a very, very long time ago. We've got someone from Regina, Canada, Cincinnati, Ohio. Fantastic. Wisconsin, all around planet Earth. Well, I think we've actually been more concentrated in one side of planet Earth, but Iraqi Kurdistan, we really are going all over the planet. That is really fantastic. So, folks, I know that I think I said that we were going to do FRQs last time. Um, and we can do FRQs. I've come prepared to do multiple choice questions. And tonight is going to be a slightly shorter one than last time. Oh, I see someone from my hometown, from Berkeley, California. That is fantastic. Though there are a couple of Berkeleys, and it's not necessarily Berkeley, California. But anyhow, so the plan is to do multiple choice tonight. I'll do the FRQs on another session. And let's just talk about what we're going to do on this session tonight. Wanted to just give you some review tips. I want to continue to coach you on what to do when you are not with me in these live sessions. Want to take some questions. Someone asked me what questions. I've kind of lost track at this point. Like we were going sequentially and then we started jumping around. So I'll just spend a little bit of time answering your questions, and then we'll do some practice multiple choice questions. That's not for you. Okay. So let's talk about some principles of FRQ practice, even though we're not really doing FRQs tonight. When you review for a big test like the AP Bio exam, the thing that you've got to be asking yourself all the time is, what are my gaps? How do I fill them? Where are the gaps in my knowledge? How do I fill them? In other words, you're not necessarily going systematically through the curriculum. What you want to do is you want to be doing activities that will show you what you don't know. And the way that you do that most effectively is by testing yourself. One great way to test yourself is by answering FRQs. The College Board is super generous about providing FRQs, and you should be queuing up those FRQs and writing responses to them, and then comparing them with their scoring guides. Now, that's a little bit hard to do. So I've got something that is also valuable. It's on my website, which is learn-biology.com. We've got enhanced practice FRQs where you type your response to a prompt. You then get feedback on your answer. It's as if I were sitting next to you. And it really is kind of like me, because I designed it. I didn't do the programming, but I designed the educational interaction. And it's as if I'm giving you feedback on the FRQ that you wrote a response to. And you spend time with our enhanced FRQs on learn-biology.com, and you will learn a lot. You will really learn a lot. And Maddie, I'm really glad that my Unit 8 helped you get a 9. And Jenny asks, are they about my FRQs? My FRQs are shorter than the College Board's FRQs, and they're kind of like pieces of them because they need to work on a computer screen. So they tend not to be as long as the questions that you'll find on a College Board FRQ, but it's equal tasks. And really the main thing, again, is can you do kind of like what I'm doing right now, which is talk freely in response to a prompt. So the prompt for me is, what am I going to tell students about FRQ preparation? I have a lot to say about it. Well, you have to have a lot to say about all of biology, and you won't know what you don't have to say until you try to answer some FRQs. So use the College Boards, use mine, and that's really what you've got to do. Muhammad is asking me if for the AP Bio test, do my videos include everything? Yeah, pretty much. I mean, I've done a very, very careful analysis of the course and exam description, and I use that as the outline on which I designed my videos. There was a gap. I didn't have anything on chi-square, but I recently produced a chi-square video. So, and it sort of depends on what version you are, because I've been working on these videos for a couple of years. But if you view all my videos, you're going to be in pretty good shape. But I do want to say something. Viewing a video is not interactive enough. And I just want to emphasize, you really got to interact with this stuff. You've got to be answering questions. You can do that at my website, learn-biology.com. And I might show you more about that later. But the main thing is you got to interact. And interaction is going to be the key to your success. Make myself a little bit smaller so I can go like this. Just tell you a little bit more about these enhanced FRQs. If you're stuck, you can get a hint. You can also study sample answers. Sign up. Multiple choice practice is a little bit tougher. I'm going to show a deck of multiple choice questions tonight, but the thing to emphasize is that the College Board has been very free at giving out past FRQs, not so generous with giving out past MCQs. They tend to guard those pretty closely. So I've drawn from a couple of sources of public FRQs. If you join me in my exam review, which you can find out more about here, this link that's on the screen right now, apbiosuccess.com/exam review. Then I just share everything that I have, the accumulated stuff that I've gathered as years for a teacher. But on these YouTube lives, I'm sticking to the publicly published stuff that the College Board is fine with the public seeing. So what are your options for MCQ practice? The College Board has sample FRQs in their course and exam description. We're going to go over some of those tonight. They also published a practice exam in 2013 when they produced a new curriculum. Those are great questions. We're going to look at some of them tonight. And then again, on learn-biology.com, I've got hundreds of AP style questions. Correct answers get praise, incorrect answers get hints and get repeated. Go to learn-biology.com to sign up. So let's think about time. It's 6:10. Let's spend about 15 minutes just like open-ended. You got a question about BIOLOGY. I will do my best to bring up a slide deck and answer those questions. So, and I'm just seeing, like Rebecca is saying, talking about some of the challenges that she's experienced taking these multiple choice questions. They are difficult. And all that I can say is, like, you've got to practice. The more you practice, the better you get. And I can show you tonight some tricks for answering multiple choice questions. Yes, you can absolutely. Jenny's asking a question. Can you say RUBP instead of writing out ribulose bisphosphate? Absolutely. ATP instead of adenosine triphosphate, DNA instead of deoxyribonucleic acid, mRNA instead of messenger ribonucleic acid. Yeah, you can absolutely use abbreviations. Yeah. And EV, do we have to know all the steps, or is it EV? I don't know. Do we have to know all the steps of glycolysis and the enzymes and stuff? Nope. Nope. You need to know glycolysis on kind of a big picture level. Like, for example, when I teach glycolysis, and in fact, if you listen to my glycolysis song, the one, glycolysis, come on, sugar for the breakdown. I walk you through 3 moments in glycolysis: investment, cleavage, and energy harvest. And those are about the things that you need to know for glycolysis. You sure do not need to know like all the enzymes in the Krebs cycle. Save that for college, and you might not even need to know that in college. And Yuri's asking for like longer paragraphs on the questions. And I would assume that this can apply to multiple choice questions or free response questions. Listen, this is I'm not gonna tell you that like this tough thing is not tough. It's tough. It's quite challenging. Um, all that I can say is the more that you practice, the better you will get to be. And I also want to emphasize that there starts to be a little bit of panic now. But if you are grown up and mature, what you will do is you will sit down with a calendar and you will mark out the hours every day that you are going to study for AP biology. And I hope that that number adds up to like 15 hours or something like that. So like 2 hours a day. And you can put in a lot of time on the weekends also. But there are people who are going to be studying a lot. And I don't want to frame it as a competition, but you just have to really put the time in for a big comprehensive test like this to get to the level of knowledge that you need to get to. So put the time in and don't like just put it on your to-do list. Say that, oh, between 6 and 8 p.m. on 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 Saturday night. Yeah, on Saturday night. You're going to study biology. And then on Sunday, you're going to do it from 9 a.m. to 1 p.m. Put in those 4 hours and then just set that time aside. It's called time boxing. And that is how you get stuff done. And just put the time in. I just heard from a student who commented on my channel on Science Music Videos that there's like an alternative test called the CLEP. And this kid went through like all 470 flashcards on my website and wound up killing it on the CLEP. And that stuff makes me feel very happy. And all that I can say again is that if you put the time in, you will get there. You will get there. So let me look at the comments. Yeah, so Blake, yeah, you're having trouble because they're hard. And all that I can say is that continue to struggle with it, continue to work with it, continue to answer FRQs that are from the College Board, and then compare that to the scoring guide. Continue to use my FRQs and look at the feedback that you'll get on my enhanced FRQs. And it's going to be difficult. It's going to feel like, oh, I don't know this stuff. But you keep on not knowing the stuff until you know it. And I see someone who's like pink Crayola marker, 51 out of 60 on the MCQs. That's unbelievable. If you're like getting 45, like 75%, you're setting yourself up for a 4 or 5. And I don't want to be more specific than that because I don't really know how the whole thing breaks down. But this ain't like your teacher's test where like, 9 out of 10 is an A. This is much, much harder. So you got to be kind to yourself. And Pinky, pink Crayola marker, 51 out of 60. You go. You go. Way to go. That's fantastic. No, I don't know how to pronounce your name, but I'm gonna do my best. Shweta. No, biomes aren't even really on the AP Bio exam anymore. That's like old. Don't memorize anything about biomes. Biomes not on the AP Bio exam. In fact, I'm not even sure if the carbon cycle is on the AP Bio exam, though it is going to be on next year's, but you don't care. You're not taking AP Bio next year. Yeah. So, and Sanjana is asking, what are some major topics that are guaranteed to show up on the AP Bio FRQs? I don't know. Nobody knows. They're going to choose 6 questions that come from the whole AP Bio curriculum. And you know what the thing to do? Learn the curriculum. How do you learn the curriculum? I've got 470 flashcards that are waiting for you. I've got a curriculum called the AP Bio Video Pathway, where I've got videos, summaries, flashcards, and quizzes about every topic. Set the time aside and start doing the work. That's what you've got to do. But I want to go back to what I said before. Test yourself. Test yourself. How do you test yourself? I've got a checklist. It's called the AP Bio, Mr. W's AP Bio checklist. Just go to, well, I think I have a banner that says this. Okay, well, if you really want to get everything, here, do this. So download my plan. My plan has links to a lot of resources. So download my plan. And another thing also is if you subscribe to my website, learn-biology.com, you'll also get on my mailing list, and then I mail out resources and stuff like that. So you get another benefit. But guys, let's do this. Let's review some specific topics. Anybody have something that they want to go over? But just put it in the chat, and if I haven't gone over it in one of my previous videos, I'm happy to take it on. So I don't want to do the Krebs cycle. I've done that before. Ecology. Okay, I got a request for meiosis. Got a request for Unit 8. Carbon cycle. Yeah. Carbon cycle, I don't want to, it's very hard to say. I would be somewhat familiar with the carbon cycle. I've got a song about the carbon cycle. And if like you listen to the song, you probably know everything you need to know about the carbon cycle. So I did water potential in the previous show. Hardy-Weinberg. Yeah, that's what we're going to do. Hardy-Weinberg tonight. So let me do this. I am going to go to here. I understand you can't see anything yet. Hardy-Weinberg. Hardy-Weinberg is in Unit 7. And I am going to stop sharing for a minute. Okay, so now you got just me, and now I'm going to present. And I'm going to go here, share screen, Unit 7. Bam. Yeah, got it. Slideshow. Let's go like this. Presenter view. Beautiful. And now I'm going to make myself small and the screen big, because that's what you need. Yeah, that's what you need, guys. And I'm going to take away that banner, even though I'm trying to promote my stuff. It's true. I am trying to promote my stuff. Let's get rid of that banner. Bam. Okay. So what we want to talk about is Hardy-Weinberg. It's all natural selection, blasting through natural selection. Okay. So population genetics is the study of how genes get distributed in populations, right? So it's looking at genes and how gene frequencies change in populations. So like, for example, here's a gene pool, totally simplified. We're just looking at one gene, big A, the big A gene. Who knows what it codes for? But in this gene, little a has a frequency of 0.5. That means of all of the alleles in this gene pool, there are 40, 20 of them are little a, 0.5 allele frequency. So that gives us another lens by which we can look at evolution. And here's how that works. A gene pool, which I just indicated, is all the alleles of all the genes in a population. You can define evolution as a change in the genetic makeup of a population over time. And the changes evolutionarily are changes in allele frequencies. Now, I want to emphasize the biggest misconception that I've been fighting against for years. And that is the idea that a dominant allele has to be more common than a recessive allele, or if it's not, it will become more common over time. And this is sort of a category error. It's just wrong. Allele frequency is based on the advantage or harm an allele confers to its associated phenotype, or random historical stuff. An example is that this is the actor Kyle Pacek, Pacek. His picture's on Wikipedia, and he's a famous actor. And he has a condition called achondroplasia, which causes some of the long bones to not grow very much. So he's a short person. That allele is exceedingly rare, but it's also a dominant allele. So that brings us to Hardy-Weinberg. There are two equations that describe how genes work in populations. And those are P plus Q equals 1 and P squared plus 2PQ plus Q squared equals 1. P represents the frequency of the dominant allele, and Q represents the frequency of the recessive allele. So what does P plus Q plus 1 mean? It means that for a gene with two alleles, the frequency of the dominant allele plus the frequency of the recessive allele equals all the alleles. So simple. If you take this expression, P plus Q, and you square it, then you get P squared plus 2PQ plus Q squared equals 1. In other words, you square both sides of the equation. Now, I'm not a math teacher, but if you square one side of the equation, you got to square the other side. This remains 1, and this is now a polynomial. And the meaning changes, and it's very important to understand what the meaning is. P squared is the frequency of homozygous dominance. 2PQ is the frequency of heterozygotes, and Q squared is the frequency of recessives, individuals with the recessive phenotype. And if you add all those up, P squared plus 2PQ plus Q squared, you get all the individuals in the population. I want to actually clear up something. I can't and don't share my slideshows, but on my website, you got my videos, you got summaries, you got flashcards, you got quizzes. So I don't give that away. I'm sorry. But that is what I do give. And I of course I'm giving this. So here's the magic way to solve Hardy-Weinberg problems that not as many people use as I would like to, because a lot of people pull out their scientific calculators and start solving for square roots and stuff like that. There's a much easier way. You've identified that 49% of the mice in a population have the recessive trait of white fur. Figure out the frequency of P, Q, P squared, 2PQ, and Q squared. Here's how you do this. If 49%, have the recessive trait, then that is Q squared. Put 0.49 in the lower right column. That was badly said. Put 0.49 in the lower right cell of your, it looks like a Punnett square. It's really better called a cross multiplication table. Now watch this, because it's so cool. It'll blow your mind if you've been struggling with Hardy-Weinberg. Here's 0.49. That's Q squared. That means that this number has to be the square root of whatever this is. What's the square root of 0.49? 0.7. 0.7 squared is 0.49. Now, if this is 0.7, we know that P plus Q equals 1. P plus Q equals 1. If this is 0.7, this has to be 0.3, because 0.7 plus 0.3 equals 1. Oh my goodness. Now check out the next move. If this is 0.3, we put 0.3 over here, and that means that now we know the frequency of homozygous dominance. It's 0.9. And once we know that, we just take this, multiply it by this, we get 0.21. And the frequency of heterozygotes is 0.42. It's 2PQ. This is PQ, this is PQ, this is PQ, this is PQ, 2PQ. So basically, the magic move when you're doing Hardy-Weinberg is to find Q squared, and it's almost always given to you in some way, and then you work from there. Timothy's asking, how come P is 0.3? Because of the Hardy-Weinberg equation. P plus Q equals 1. Therefore, if Q over here is 0.7, then P has to be 0.3 because P plus Q equals 1. Jenny, that was a beautiful comment that you put in there. So anyhow, that's exactly it. So this is the solution to like 99% of your Hardy-Weinberg problems. And you can take it further. The frequency of the dominant phenotype is the homozygous dominance plus the heterozygote. So it's 0.9 plus 42, 0.42 equals 0.51. That's it. So that'll get you through 90% of your Hardy-Weinberg problems. Now, there's another thing that's called the Hardy-Weinberg principle. And that's more theoretical. It's not solving mathematical equations. Excuse me while I take a sip of tea. And I'm looking at Cheesy coli, which is a fantastic name that has a lot of biology in it. How do you know whether to square a number they give you in a question? I'm just going to go back here for a sec. You have to read the question. And in the question, you have to figure out, like you're asking yourself, what have they given me? Have they given me P? Have they given me Q? Have they given me Q squared? If they've given you P, if they tell you like, oh, in a population, the frequency of the dominant allele is 0.7, well, then you just, I'm sorry, I should have said 0.3 to accord with this over here. It's 0.3, then it's like, oh, if this is 0.3, then this has to be 0.7. And then you would square this to get Q squared over here. So you just have to like get oriented and figure out what's P, what's Q, or what's Q squared, and then you'll figure it out from there. And I'm just checking the chat, and people are just greeting one another. There's a beautiful little community thing happening here. So the Hardy-Weinberg principle says that the frequency of alleles in gene pools will stay constant unless one or more of the following conditions is not met. Not met. So it's kind of like a little bit of negative logic here. So basic idea. Gene pools won't change. Their gene frequencies won't change unless something happens. What are these conditions that constitute a non-evolving population? The population has to be infinitely large. How could that be? We'll get there. The population has to have no harmful or beneficial alleles. The population has to be one in which there's random mating. There may not be any emigration, people leaving, taking their genes with them, or immigration, bringing in new alleles. And there can't be any net mutation of one allele into another. You're probably looking at that and you're saying, Mr. W, how can that be? There's no population like that. That's exactly the point of the Hardy-Weinberg principle. If these conditions are violated, then populations do change. Here are some factors that cause evolution. Genetic drift. If a population is small, you can have random changes in allele frequencies. If there's natural selection, that means that there's either a harmful or a beneficial allele. The allele frequencies will change. If there's sexual selection, if certain phenotypes are more sexually desirable in a sexually reproducing population, you'll have change in allele frequency. If there's gene flow, alleles coming in, alleles going out, you'll have gene flow. And if there's a tendency of one allele to mutate into another. Okay, so folks, I think that's about as far as I should go with, except for one idea. Okay, genetic drift. So genetic drift is Random change in allele frequencies in a population's gene pool, usually associated with small population size. Think about yourself drifting on a raft, like you'll randomly move over to one side of the pool or the other. You're not doing it with any direction. Well, this is the same thing. If you were to just randomly pull out some alleles from a gene pool, you can wind up with a sampling error that changes the gene pool. One kind of genetic drift is called a population bottleneck, and that means that either a biotic, a living, or abiotic non-living factor wipes out a large percentage of the individuals in a population, leaving only a few survivors, and the survivors' alleles might not be representative of the larger population. So in this case, we're showing a population that goes through a bottleneck, sort of goes through a funnel, and in this case, all of the capital B alleles are lost from this population. Some alleles might disappear completely. And note that the survivors didn't have any selective advantage. They were just lucky. This isn't selection, it's drift. Another kind of genetic drift is called the founder effect. And that's a type of genetic drift. And it's when a small number of individuals from a large population found a new population. And because only a small number of individuals come, the allele frequencies in the gene pool of the founders might be different from those in the parent population. A kind of famous example of that did not show up in this slideshow. I wonder what happened. I lost an image. But there's a condition that is common among the Pennsylvania Amish, and it often involves, well, I don't need to actually say more because it's a little bit of a stigmatizing thing. But among the Amish, there are different gene frequencies than among the surrounding populations. And that's really all that needs to be said. So let's stop there. And I wanted to go over some multiple choice questions, and let's go and do that. And I wonder if I can just go like that. Yeah, good, good. So, not to repeat the stuff that we did at the last session, we're going to jump. No, too far. Here, fantastic. So, this should be all new stuff. And we're going to play a game. And the game is going to be answer the question, but we have to play it in the right way. I'm going to present a question, and then when I say send, you're going to send your answer. And it's easy because it's like A, B, C, or D, right? You're just going to send your answer. But if you do it like as soon as you see it, you're going to ruin the experience for everybody else because it's hard to like think when you already see the answer. Okay, so let's start. I might ask you to do some reading silently as my throat becomes more and more labored from this kind of incessant talking. Different photosynthetic organisms have different types of chlorophyll molecules. The distribution of chlorophylls in several different groups of organisms is shown in Table 1. A plus sign in the table indicates the presence of a chlorophyll, while a minus sign indicates its absence. So here we see for flowering plants, green algae, brown algae, red algae, and cyanobacteria, what type of chlorophyll they have. And that's indicated with a plus or a minus. Based on the data, which of the following most likely describes the evolutionary relationship among the organisms? I'm going to read this silently. You read it silently. When I say send your answer. Okay? So everyone's reading these choices. Okay, now don't send. People have expressed some difficulty dealing with multiple choice questions. I've written some multiple choice questions, and they're hard to write because you need to lie. What I mean by lying is like every wrong choice is a kind of lie. It's an untruth as opposed to the one true statement. So, one of your best friends is the process of elimination. And you can eliminate based on a couple of criteria. One of them is there no evidence for this? So, let's look at these choices. Because brown algae, red algae, and cyanobacteria lack chlorophyll B, they evolved before green algae and flowering plants did. There's no evidence for that here. So I would kind of just like put a question mark next to A and say, oh, I don't really think about that. B, because green algae and flowering plants contain chloroplasts, they evolved more recently than brown algae, red algae, and cyanobacteria did. Well, first of all, brown algae and red algae have chloroplasts. So that doesn't look good. So another criterion is like bad biology. Now, you might not know that. I know because I'm a biology teacher. Um, C, because increasingly complex forms of chlorophyll are found in red algae, brown algae, green algae, and flowering plants, respectively. This reflects the order of their appearance. There's no evidence about complexity of chlorophyll. So far, we've got three very dubious answers, which makes us think that D is probably right. And D is because all the organisms contain chlorophyll A, the organisms share a common ancestor. And that is the answer. The answer is D. And this shared um, trait, this is called a shared derived character. And that indicates that these organisms constitute a clade and they're united by this shared derived character. So here's something kind of surprising. This question is actually not about photosynthesis. This is a question about phylogeny. It belongs to unit 7. So I just put the word in the chat. And what you might want to do right now is like grab a piece of paper or, probably your phone is not really a good thing to have out right now. But you probably have it. But the question, what you might want to note is that if you had problems with this, you got to study phylogeny because in phylogeny, we use trait tables to figure out evolutionary relationships. That's what this question is about. Oh, Ah, I'm going to skip this question and I'm going to come back to it. Kind of did it again. All right. All right. The human TPM1 gene encodes members of the tropomyosin family of cytoskeletal proteins. Which of the following best explains how different proteins can be made in different cell types from the one TPM1 gene? From the one TPM1 gene. So you've got 4 choices. I'd like you to silently read those choices. And then when I say send, what I'd like you to do to help me out so that I know what the comment is, put 11 dash and then answer A, B, C, or D. So everybody read the question, A, B, C, or D, and then we'll look at it together. Okay. Read silently, silent reading period. No talking. Luke, did I say send? I didn't say send, but everybody send. Go ahead. Send your answer. Okay. Want to show you a picture that's really going to solidify this. I'm going to go to Learn Dash Biology, sharing this tab. I'm going to AP Biology. I'm going to Unit 6. I'm going to Eukaryotic Controller Transcription, and I'm going to show you this fantastic picture. Okay, so this is alternative splicing, and that's what's going on here. Eukaryotic genes have exons and introns. Exons are the expressed sequences. They become protons. I'm obviously having some trouble. They become proteins or parts of proteins. The introns are intervening sequences. They get cut out, spliced out. So what happens is, here's the DNA. The DNA gets transcribed into pre-mRNA, which still contains the introns. Then there's a team of enzymes, many of which are RNAs, that splice out the introns and connect the exons. But they don't always connect all the exons in order. There's alternative splicing where, like, for example, this variant of the protein has exon 1, 3, they skip 2, 4, they skip 5, and they do 6. This variant of the protein has exon 1, 2, 5, and 6. And this one has exon 1, 2, 4, 5, and 6. And you'll notice that the shape of all of the proteins in this family is subtly different. So that's alternative splicing. It's a super important concept to know about. And we're going to go back here and I'm going to share this tab. And that is why the answer to this question is B. Different exons are retained or spliced out of the primary transcript. Okay, now, so that I can show that other question that I like so much. Oh, look at how small I am over there on the sign. It looks terrible. But I'm going to go like this, going to go like this. A little trouble here. Not sure why. There we go. Okay, so now I'm going to share screen, share screen. Fantastic. Okay, so at this point, nobody remembers what the answer was that I inadvertently flashed on the screen. And this is an important question because it talks about another topic. By the way, the previous question, if you got it wrong, you got to review eukaryotic gene regulation and alternative splicing. So now let's check out this. This is from a lab that I hope you got to do. A student used a microscope to observe a wet mount slide of a red onion epidermal cells that were suspended in a 1% NaCl solution. The student then added a 15% NaCl solution to the slide, and observed the changes that occurred. The student's observations are represented in figure 1. Here's figure 1 right here. So this is 1% NaCl. This is 15% NaCl. Which of the following most directly explains the changes in the cells? Folks, look at those answers. When I say send, send it, but don't send it yet. Everybody look at the 4 answers. And then when I tell you, you're going to put 10 dash whatever the answer is. 10 dash, send it, folks. What's the answer to this question? Great job, folks. Great job. The answer is indeed C. This question is about osmosis. I have a song about osmosis that actually references this very lab with a different organism, not with onion, with a kind of aquatic plant. So here's the deal. When you put this 15% salt solution, you've created an extremely hypertonic environment. Water always flows from hypotonic to hypertonic, and water will flow out of the cells. The vacuole holds most of the water, so the vacuole kind of shrivels up, and that brings the membrane along with it. So this is basically the cell membrane surrounding the cytoplasm, and now there's sort of empty space inside the cell. So the answer indeed is C. If you got this wrong, then you're trying to track what you need to study. You're trying to find your gaps and fill them. Fill the gap related to osmosis, okay? And remember, you should say this in your sleep. Water flows from hypotonic to hypertonic. Okay, we just did this question. Okay, now, love this question. Very cool question. Scientists examined the folded structure of a purified protein resuspended in water and found that amino acids with nonpolar R groups were primarily buried in the middle of the protein, whereas amino acids with polar R groups were primarily on the surface of the protein. Which of the following best explains the location of the amino acids in the protein, in the folded protein? Folks, silently read A, B, C, and D. And I'm going to do the same to sort of time it. And then I'm going to say send. You're going to put 12 dash and then the answer. I'm reading A. Now I'm reading B. Now I'm reading C. Now I'm reading D. Folks, you may send 12 dash with an answer, A, B, C, D. Okay, folks, you're doing good. All right. The answer indeed is C. I'm going to go show you another great picture. Um, where are we going to go? We're going to go to here. We're going to go to AP Bio Unit 1. We're going to go to protein structure. And this is good because it's going to give us a little opportunity to review protein structure. And we're going to go all the way down here to tertiary structure. All right. So let's see if I can get this to be bigger. No, but I can go like this. That works pretty well. Okay. So here's a diagram that shows the tertiary level of interactions that form protein structure. So one is the polypeptide backbone. So carbon, carbon, nitrogen, carbon, carbon, nitrogen. And this shows four kinds of interactions between R groups. This, for example, is a hydrogen bond. This is a disulfide bridge, which is a covalent bond. And what do we have here? We have a bunch of hydrophobic functional groups that are all clustering together on the inside of the protein. They're sort of avoiding water because they don't form hydrogen bonds with it. This protein structure, got to know it. So if you got this question wrong, in your little pad of paper where you're keeping track of what you need to study, you need to study protein structure. And I've got a tutorial on it, but I'm trying not to sell too much. Okay, so let's go back to here. Let's go back to here. Answer was C. Let's do another question, folks. Oh, love this question. I'm just looking at the chat. Okay, we're okay. People are complaining about their teachers a lot. Sorry. Anyhow, the apple maggot fly, Rhagoletis pomonella, is native to North America and originally fed on the fruit of the wild hawthorn. Since the mid 1800s, a population of flies has emerged that instead feed on domesticated apples. Apple maggot flies typically mate on or near the fruit of their host plants. Many varieties of apples ripen 3 to 4 weeks before the hawthorn fruits do. The different fruit preferences of the two fly populations will most likely have which of the following effects? Folks, silently read A, B, C, and D. I'm going to do the same. When I say send, put 13 dash with a letter. Folks, I'm done reading, and I would like you to put the number 13 with a letter A, B, C, or D. Send it. Okay. I'm watching the answers come in. Okay. The answer is B. This is a setup for speciation because what we've done is we've created a barrier between these two populations, allowing each of them to evolve differences because of natural selection and genetic drift. Now, I have a follow up question. First follow up question, I'm going to put it in the chat. Is this pre or post zygotic? Okay, so I just put a question in. I'm Science Music Videos. Is this a pre-zygotic or a post-zygotic? Tell me what it is. I'm seeing some answers come in. You can just put pre or post. Good. Okay, now let's get to the AP Bio 4 or 5 level. What kind of pre-zygotic isolating mechanism? So I'm just putting in what kind. What kind of pre-zygotic mechanism? Andrew. Okay, Nikhil. Okay, so let me clarify a couple of things. Pre-zygotic means that it's a barrier that happens before the formation of the zygote. These two populations of flies are never going to get to interbreed, therefore pre-zygotic, right? Before the formation of the zygote. In terms of the kind of barrier, a behavioral barrier is one where the behavior of one organism is essentially a turnoff to the other organism. It's like a mismatch in terms of courtship or something like that. So, like for example, one kind of sparrow sings a song that another kind of sparrow says, that's a weird song. I am not going to come close to you. Or, birds have all these incredible like courtship dances and stuff like that. And like the dance looks strange. And no, I don't like that. So anyhow, that's what behavioral barriers are. This is a temporal barrier. It's based on time. Based on time. I just also want to say something. Remember this whole idea of process of elimination? The ability to survive on a diet of two different fruits will help the flies learn to eat many more types of fruit. You could tell that the writer there was just like struggling. Like they couldn't really think of something good because flies learning, I don't know. Do they learn? Maybe they learn a little bit. I don't know. I don't want to be anti-fly. Okay. No, ruined it. Okay, we're just going to skip this question. All right. So we're just going right to 15. Sorry, I clicked too much. It is estimated that oxygen production first evolved in photosynthetic prokaryotes approximately 2.7 billion years ago. The first photosynthetic prokaryotes are presumed to be similar to today's cyanobacteria. Which of the following best supports the claim that photosynthetic prokaryotes were responsible for oxygen in the Earth's atmosphere? Folks, read it. A, B, C, and D. I'm going to do the same, and then I'm going to say send 15 dash something. Okay, I've had time to read it. Hopefully you have. Folks, put in 15 and then an A, B, C, or D. Let's see it. Okay, I'm seeing a split between A and B, which means I got to do a little review. This question is a photosynthesis question. I know it's sort of got an evolutionary twist to it, but it's really about photosynthesis. The previous question was a speciation question. So let's go to where I got the diagrams. Let's go here. Good. I'm showing the right thing. We're going to go here, 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 and here. Okay. So this is a very conceptual diagram of, I think that's about as big as I can make it. Yeah, this is a very conceptual diagram of photosynthesis. And I organize on my website a lot of diagrams like this because I don't, well, I guess I did label the one on the right. So silly me. So basically, here's the deal. Photosynthesis has two major moments. On the left side, I'm showing the light reactions. On the right side, I'm showing the Calvin cycle. And this organelle is a chloroplast. That's where all the action is happening. Water comes in, and during the light reactions, it's split apart. It's split apart into protons and oxygen. The oxygen bubbles away. If you were lucky enough to do the spinach leaf disc lab in class, that's what it's all about. It's all about those spinach leaf discs producing oxygen. So the key thing, and this is widely misunderstood, the source of the oxygen in photosynthesis is water molecules that are split apart. And then the products of the light reactions, which are the electron carrier NADPH and ATP, feed into the Calvin cycle, which is used to take carbon dioxide and reduce it, chemically reduce it into sugars. Now I'm just going to jump, not here, going to jump again. Here. Okay, so right here, this shows the light reactions, and you can see that light is hitting a photosystem. It's hitting photosystem 2, and photosystem 2 is taking a water molecule and splitting it apart into oxygen and protons. It's also taking electrons from water, and those electrons are going into an electron transport chain that's being used to pump protons and ultimately to reduce NADP plus into NADPH, and then later through a process that is unbelievably similar to what happens in the mitochondria during electron transport chain. ATP synthase is used to create ATP from ADP and phosphate. Folks, I can only do an hour. So I just want to say, sign up for learn-biology.com. It's going to totally change your experience as a biology learner. If you want to cram during the next, no, don't cram. If you want to get ready to learn more than you ever thought possible, use learnbiology.com to make it happen. And you'll just be so happy. I just want to also encourage you to not only sign up for learn-biology.com, but also I'm doing a whole bunch of Zoom review. Those are paid events, but check it out. And if you have the means to do it, it's going to be super valuable. So let's just say goodbye. If you could just put into the chat, say, thanks, Mr. W. Oh, where's my schedule of these? Live reviews. Oh, yes. Oh, fantastic. So here's this. I'll put this in the chat. So here, live reviews. Today's Thursday. So my next session, it's a paid session. It's on Sunday, but then I'll be back on YouTube Monday night. All right. So, folks, you're the best. I totally admire you. Put some love in the chat. Just say thank you. Um, and yeah, love seeing that. Thank you. Good, good. Appreciate that. All right. You guys don't need to pull all-nighters. What you need to do is organize yourselves to study slowly, carefully, and do it. No need for all-nighters. Folks, you're the best. This was fun. I'll see you at the next live event, which is going to be Monday. I hope I see a bunch of you on Sunday. Sign up for Sunday. You won't believe how much you'll learn. Everybody, be well, be safe. See you soon. Learn, Biology.